All right, welcome back to our third video for Chapter 2. We're going to continue Section 2.3 here. At the end of the previous video, we started talking about different types of light microscopy, starting with bright field. Remember, bright field was when we saw darker images against a bright background. In this case, our second type, dark field microscopy, we're going to see the opposite. Now we're going to see bright images against a dark background. And dark field microscopy is actually a modified version of bright field microscopy. And I'm going to talk about these parts in the next slide. Here's what the dark field microscope would look like, with this being the bottom or the lamp or light source. Sometimes we also call that an illuminator. Here we would have our condenser area, condenser region, which has the condenser lens, and usually we have something called a spinner or a condenser turret, where we can change the settings from bright field to dark field. Within that condenser or condenser turret, we have something called an opaque disc or opaque light stop. Sometimes we just call it a light stop, which basically blocks most of the light coming from the light source, but allows the edges to go through, and you can only see part of this. This is the cross section. So kind of a ring of light will pass through, allowing a hollow cone of light to pass through the specimen. If there were no specimen, the light would continue to move as like a cone, and nothing would be captured by the condenser. It would just, everything would appear dark. But if you have a specimen and the specimen is on here and scatters or reflects or refracts the light, then it will be bent such that the light will enter the objective lens. The resulting finding, or what you see through the microscope, is now bright objects against a dark background. Our book provides an example here of an image of Treponema pallidum, the bacterium that causes the sexually transmitted infection syphilis. And you can see clearly that we see bright cells against the dark background. So dark field microscopy is great for viewing live specimens that can't be stained, right? They might be damaged if you try to stain them. So they're good for looking at thin, delicate, live specimens, but the drawback is you can't really see internal structures, and we'll use a different kind of microscopy for that. Here's a checkpoint to look at differences between bright field and dark field. So if I think about the illuminator or the light, remember in bright field there's no stop, so light just goes through the specimen. There's no blocking of light, whereas in dark field we use that opaque disc or that light stop to block direct light, right? Only scattered or reflected light will reach that objective lens. Looking at the background, for bright field, remember the specimen is going to be dark against a bright background, whereas in dark field it's the opposite. The specimen will appear bright against a dark background. For bright field, we usually need stains to increase contrast, so you can see the specimen against the background. For dark field, we're not staining because we're looking at live specimens. And so again, same idea for applications. Bright field, we use stained, fixed samples, so we've treated them. They're usually dead. In dark field, we're viewing live, usually delicate, unstained specimens, just like the ones they showed us in the book, Treponema pallidum. Here our book brings us back to that clinical focus problem. Here, Cindy's sample is going to be sent to a medical lab that's pretty small and poorly equipped, so they do what they can, including making a wet mount from the sample. Here they're just putting the sample on a slide and trying to check it out under bright field microscopy, but because cells are transparent pretty much against the background, you can't see much. So to increase contrast, the lab tech inserts an opaque stop. So we're doing dark field microscopy. And now with dark field microscopy, she can see, or he can see, that the cells are spherical and grouped in clusters. So think about why shape and growth pattern, that cluster, might be important, and then what other kinds of microscopy could be used. Our third type of light microscope are the phase contrast microscopes, which are going to take advantage of the bending of light and wave interference. Remember, there was constructive and destructive to generate high contrast images. we're going to see they're kind of similar to dark field microscopes in that they're not going to require staining. And a benefit of phase contrast compared to dark field is that you're going to be able to see internal structures even better. The rest of this I'm going to talk about using the next image. Here's the image of the phase contrast microscope from our book. So here we would have our condenser with a condenser lens, and here we have something called the annular stop or annular ring. The side view is hard to tell what it's doing, but here's the top view. Only light from the light source that passes through this area, this ring, can go through. So you'll end up producing a hollow cone of light through your specimen, similar to dark field microscopy. The difference here in phase contrast microscopy is that if this whole thing were our objective, one of our objective lenses, there would be a lens here, but also a phase plate or a phase ring here. And it's hard to see from this image, but a phase ring would look like this, where light from the specimen can either go through this region of the phase ring or this region of the phase ring. And this will result in waves that are out of phase. So the waves can go through constructive interference or destructive interference. For example, when the light waves are out of phase, they'll cancel each other. And so ultimately, when the specimen is able to bend or refract the light, it's going to appear darker against a bright background of unrefracted light. And structures in your specimen that have different refractive indices will differ in their different levels of darkness. Here is another look at the bottom half of that previous image. Here's that annular stop, producing the hollow cone of light through your sample. And then here's the sample. So pretend you have some kind of specimen on your glass slide. Remember that, let's say this were some cells. We have all sorts of structures within the cell. We have the lipid membrane, maybe a nucleus, maybe different proteins. They're all going to bend light in different ways depending on their individual refractive indices. As the light bends in different ways through the specimen, they're going to pass through different areas of the phase ring within the objective lens, and ultimately you'll see waves that are in phase or out of phase that reach our eyes. If they're in phase, remember if two waves are in phase, they're going to go through adding or constructive interference, and you'll see brighter, usually brighter images. If two waves are completely out of phase, you can see this is going through a peak while this is going through the trough, you'll see negative or destructive interference, and that leads to dark images. Going back to our textbook, here's the condenser with that annular ring or stop. That's going to produce a cone of light that goes through our specimen, and our specimen is going to refract or reflect light. Light traveling directly from the condenser versus light traveling through the specimen are going to be out of phase when they enter the phase plate and that condenser lens. Wavelengths that are in phase are going to add together. If they're out of phase, they'll cancel each other out. These come from a different site, but I like the image of the condenser turret. We have something similar in our microscopes that allows you to change the condenser settings. And here you can see bright field position, as well as different types of annular stops with different diameters of their cones of light that will pass through the specimen. This site also has a kind of virtual phase contrast microscope where you can change the settings and look at how it affects your image. So phase contrast microscopy is great when you're looking at live specimens. These are see-through or very low contrast. We can also use it to view organelles and eukaryotic cells, as well as specific structures in prokaryotic cells like endospores and bacteria. So we use phase contrast usually when we cannot stain it, when we cannot stain the organism because it'll damage or kill it. So great to use phase contrast for live unstained samples. We can also use dark field, right? Dark field can also be used for live specimens. The main difference is that for phase contrast, that's better for looking at internal structures or specific cell structures. Whereas dark field, you can only really see the edges or outlines of the cell, of the organism. Here's an image comparing bright field versus phase contrast microscopy, and you can see much more detail in phase contrast, including the nucleus as well as other structures. Our next type of light microscope are our fluorescent microscopes. These use fluorochromes or fluorescent chromophores to show us something, to highlight something. So these chromophores, remember, they are going to absorb higher energy short wavelength light like UV or blue light and emit lower energy longer wavelength light, some kind of color we can see. So examples of these chromophores include Texas red or DAPI. You might see others in your reading. And they're going to end up producing colored images against a dark background. These are very helpful in both clinical micro and cell bio to help us either identify a pathogen is present, look at specific structures or molecules, or even differentiate between living and dead cells. We can even use multiple different fluorochromes on the same specimen to help label different features of that specimen. We're going to learn more about antibodies when we get to the immune system, but in summary, antibodies are proteins that kind of look like this, that are very specific in what they will bind to. Each antibody will only bind to a specific structure called its antigen. And immunofluorescence takes advantage of this, in that you can make immunofluorescent antibodies that bind to a target structure. For example, you can use these antibodies to see if a pathogen is present or to highlight specific cell parts, like the cytoskeleton of a cell. There are two types. We have direct immunofluorescence, which the chromophore is attached to the primary antibody, or we have indirect immunofluorescence, in which there's a secondary antibody that's labeled, and that's going to bind to the unlabeled primary antibody. Here's another look at direct immunofluorescence. Here is the primary antibody, and we can see it's directly labeled with a fluorochrome. So anything that the primary antibody binds to will show up as a brightly lit structure, some kind of colored structure. Here is indirect immunofluorescence. The primary antibody is the black one, and that's not labeled. If you only had that one, you wouldn't see anything under the microscope. But here we have a secondary antibody that does have a label, has a fluorochrome. So if the secondary antibody binds to the present primary antibody, you'll see the color appear. And this allows for signal amplification, indirect immunofluorescence does, because you can have multiple secondary antibodies bound to the primary antibody. Our picture in the book doesn't show it, but you could have another one here and another one here. And if you have all three bound to the primary antibody, the signal would appear 3 times as intense than the single one over here on the left. The next type of light microscopy we have here is confocal microscopy. And instead of using a lamp or a broad beam of light, we're going to use a laser for confocal microscopy. It scans multiple Z-planes, which are depths, in a specimen. So let's pretend I have a fake specimen here, like a cube. It's a three-dimensional specimen. What the laser would do is scan one depth at a time. So let's say I scan that surface, and then I'm going to go deeper into the specimen and scan the next layer, and then maybe even deeper, and so forth. So what I get are several or many two-dimensional high-resolution images that are going to be reconstructed into a three-dimensional model by a computer. So this is great for observing things that are thick while alive as well. So we can look at things like biofilms. In some cases, you might use confocal microscopy in combination with fluorescent chromophores to enhance the contrast and resolution. And our book provides a link that allows you to see an image generated through confocal microscopy. And you can see this three-dimensional image here. Now we're going to switch gears and look at electron microscopy. This is not a type of light microscope because we're going to be using short wavelength, high energy electron beams instead of light. This allows us to get extremely high resolution. Remember that electrons have very small wavelengths, up to about 0.005 nanometers compared to visible light, which was about 380 to 700 nanometers. So that means it's much shorter wavelength, much higher energy. This allows us to magnify up to about 100,000 times usually, sometimes perhaps even up to 2 million times the original size of the structure, which allows us to see subcellular structures, so things within the cell like the mitochondria and even perhaps DNA strands. This also allows us to see viruses that we could not see under typical light microscopes. One of the drawbacks is that you cannot use electron microscopy on living specimens simply because of how we prep the samples. So these can only show dead specimens or structures. Here's a picture of a transmission electron microscope from our book. The sample usually goes here. Prior to that, you actually have to prep your sample on this tiny little disc that, if you were to zoom in, kind of looks like this. So your sample is there, and you'll see tiny little grids here. This can be zoomed in on using the computer, and you're going to basically look through each of these grids for your sample of interest or specimen of interest. In our class, we're going to skip the mechanics of electron microscopy, although I do want you to realize or remember that we're using electrons instead of light for electron microscopy. This gives us much greater magnification and resolution due to the much smaller wavelength of electrons. And also very briefly, I'd like you to know the two major types. One is transmission electron microscopy, which requires you to cut your sample into very thin sections, and electrons will pass through these thin sections to show internal structures, as shown here. Whereas the second type, scanning electron microscopy, shows you the surface of your specimen, and this produces three-dimensional detailed surface images like the one shown here on the right. Another checkpoint, let's look at some advantages and disadvantages of electron microscopy. So we have a lot greater resolution and magnification compared to light microscopy because electrons have much shorter wavelengths. We can see things like subcellular structures like organelles. We can see DNA strands. We can see viruses. For disadvantages, remember that you cannot view living specimens. It's really the preparation that I didn't go into detail about. You have to dehydrate your sample. You're going to have to fix it, stain it, cut it into really thin slices, and coat it with different metals. And the inside of the electron microscope is a vacuum, so no living specimens can be used here. It's also extremely expensive compared to light microscopy. When we're looking at TEM versus SEM, remember transmission electron microscopy is good at looking at internal structures of cells like organelles or maybe the cell membrane, and you need really thin sections, whereas scanning electron microscopy is good for looking at the surface of a specimen. We'll see biofilms again later on in the semester, but I wanted to talk about them briefly because I mentioned them in a previous slide. Essentially, a biofilm forms when a few cells attach to a surface and they start secreting basically a slime or an extracellular matrix and signals to attract even more cells to the area. They are found in nature, they're found on things like medical devices and of course in our human body like the teeth. They are notable because they can resist the immune response and even resist certain antimicrobial drugs. Because of these characteristics, we're interested in studying biofilms under the microscope as well. Some types are limited. For example, thickness is not good for light microscopy. Confocal works pretty well, and fluorescence can be used to identify specific parts. Electron microscope is not that great because it requires dehydration, and we can even use fluorescent beads to track specific parts or components of the biofilm by attaching fluorescently labeled beads to specific structures we're interested in seeing. The last type of microscopy we'll talk about is scanning probe, and in this case, it works how it sounds. We use sharp physical probes instead of light or electrons. This gives us extremely high magnification, up to 100 million times compared to the original structure, and we can even see individual atoms at this level. So we don't usually use atomic structures to diagnose anything, so it's primarily used in research rather than diagnostics. And there are two main types: scanning tunneling microscopy or microscope versus the atomic force microscope. And for our class, we're not going to learn about the details for these two. Another quick checkpoint, which has a higher mag. We just learned the scanning probe microscope has much higher magnification, right? Up to 100 million times compared to the light microscope, which is about 1,000 times. An advantage of scanning probe microscopy is that you can see individual atoms on a surface, so super high resolution, but a limitation could be that, we don't really use it for diagnostics. It's usually only used for research. The end of this section covers the different types of light microscopes. Don't worry about the ones that I did not cover. This one as well, but make sure you go over the chart and understand the basic differences. Since electron microscopes use electrons instead of visible light, we can have much greater magnification and we can see subcellular structures or surface details. Don't worry about these details, but remember that scanning probe microscopes use probes and they can magnify even greater than electron microscopes where we can see atomic level detail. All right, gather your notes and make sure you can answer these learning objectives. Take this in combination with your lab handouts, especially that microscope handout that you get in lab, to look at the different parts of a bright field microscope. Can you tell me where and what the ocular lens does? Where are the objective lenses? Where's the stage? What are the XY knobs? What is the condenser? What's the rheostat, for example? For the second one, calculate total magnification. For a compound microscope, in our lab we have two lenses. We have that ocular lens, so we take that magnification multiplied by the objective lens magnification, and when you multiply those two, you get your total mag, right? So if you have 10 times ocular lens, a 40 times objective lens, then you get 400 times total mag. For the third one, looking at the different types of light microscopes, electron microscope scanning, probe microscopes. Light microscopes, we had the bright field microscope, dark field, phase contrast, fluorescence, confocal. And I didn't talk about differential interference contrast or two photons, so don't worry about those details. If you're interested, we can talk separately on this. We also had electron microscopes, which are more powerful because electrons have shorter wavelengths. And then those scanning probe microscopes that have atomic level views. That takes us to the end of the third part. In the last section of Chapter 2, we'll look at staining.